Hi guys, alright, this is lecture 9. It's excretion organs, ammonia, uric acid and urea, or urine, urea and clever insects. Alright, these are the learning outcomes, or what I want you to be able to do by the end of the lecture. You should be able to outline generalised and specialised excretory organs and the representation across the phyla. Describe the different excretory organs utilised by invertebrates. So we're typically talking about nephidia, antennual glands and malapighian tubules. Describe the structure and function of the parts of the vertebrate kidney. Section 4 is homework, and Section 5 you need to be able to distinguish between aminotelic, uretelic, ureatelic forms of nitrogen excretion and how different organisms use them and why. So what we're really going to look at is how organisms excrete and package their nitrogenous waste, how the vertebrate kidney works, and the structure and function of other nitrogenous excretory organs. So before we start, there's a couple of keywords. We've got ammoniotelic, ureotelic and ureatelic. So looking at the words, one must be excretes ammonia, one must be excretes urea, and one must be excretes uric acid. We've got ultrafiltration and the loop of Henle. So if we're talking about ultrafiltration and the loop of Henle, we're talking about the vertebrate kidney. So these are the relevant chapters in the textbooks. And there are two papers I suggest you have a quick flick over. The first is a review looking at nitrogen excretion, and so how organisms excrete nitrogenous waste in different forms. And the second is actually by the wife of Smith-Nielsen, so Bodil. So she's the one on the left in the picture, not the pretty one on the right. And what she does is she looks at how excretion, or the excretory systems, are actually greater than the sum of parts, or how in physiology organ systems can come together to actually perform functions which they look like they should be able to. Alright, so we're talking about excretion. Okay, so there are three macronutrients that we have to consume. These are carbohydrates, fats or lipids and proteins. And what are the waste products from each of these? So before you move on to the next slide, all three of them have waste products which are common, and one of them has a waste product which the other two don't. So, thinking about what the composition is of the macronutrients, what are the waste products of these macronutrients going to be when we break them down? Alright, so here we have our three macronutrients. So carbohydrates are made of carbon and hydrogen, fats or lipids are made of carbon and hydrogen and oxygen. Sorry, our carbohydrates can have oxygen in as well. Okay, and we've got proteins which are carbon, oxygen, hydrogen and nitrogen. So carbohydrates, fats and proteins will all be broken down to produce carbon dioxide, or respire to produce carbon dioxide of water. However, when we break down proteins, we produce an extra waste metabolite, and those are nitrogenous wastes. So we start off with a protein, we break it down into amino acids, and then when we break down amino acids we produce ammonia, okay, or NH3. But there's probably going to be a problem with ammonia, and before you move on, what is the problem going to be with ammonia? Well, the problem with ammonia is that it's highly toxic, okay, or corrosive I should say. So here we've got someone who's got an ammonia burn on the bottom, and here we've got what's familiar to anyone who's a parent is nappy rash on a baby's bottom, and that's basically because they sit in sodden nappies, so the ammonia irritates the skin. So if we live in water, ammonia is not such a problem because it's highly soluble, so it'll just be diluted out by the water that surrounds us. But if we're a terrestrial organism, water's limited, okay, so there's going to be a problem with having too much ammonia in the body. So how are we going to deal with this ammonia that we produce from the breakdown of proteins? Well, the solution is simple. If ammonia is toxic, convert it into something that's less toxic. So, if we live in water, we're going to be ammoniotelic, okay, so we're ammonia excretors. But when we start looking at terrestrial systems, we see organisms which are ureotelic and ureatelic. So, why do you think some organisms might be ureotelic and some might be ureatelic? Why aren't they all ureotelic, or why aren't they all ureatelic? Well, if we look at this slide here, we can see there's a trade-off between the habitat you live in, or the amount of water that's available really, and the cost of repackaging your nitrogenous waste metabolite. So if you produce ammonia, it doesn't cost you any energy whatsoever because it's just the end breakdown product, but you need to live in water. You could convert ammonia into urea. Now urea is still toxic, but it's not toxic in low concentration, so providing you have still plenty of water to dilute it by, you're okay. So if you're a terrestrial organism living in a temperate environment where there is plenty of water, okay, then you're going to be ureatelic. However, if you're somewhere where there isn't very much water, or you want to reduce the amount of water that's in your body for some reason, because you want to reduce weight as in birds, then you're going to produce uric acid. And then if you're somewhere that's really, really dry, where you've got to conserve water, conservation of water is really important, so you're living in a desert, then you'll convert your nitrogenous waste into guanine. However, as you move from ammonia to urea to uric acid to guanine, it's costing you energy each time to make the nitrogenous waste safer, but actually what you're doing is you're taking a simple compound and making it more complex. So the majority of scorpions and spiders, they will excrete 90% of their nitrogenous waste as guanine. And interestingly, guanine is responsible for why fish scales are shiny and why for lining the inside of fish swim bladders. And this is because guanine, and once it's laid down as a crystal, is actually impermeable. Other thing to note here is that actually, organisms don't, they won't be particularly just excrete urea. They might excrete urea, some uric acid, and some guanine. So there's a mixture of strategies, and what you will excrete will depend upon your normal environment. So if we look here at the bullfrogs and what they excrete as they metamorphose from a tadpole into a frog, we can quite clearly see that as they move through different life stages, what they excrete changes, and that would make sense. So if you're a tadpole, you're going to excrete ammonia because it's cheap and you're in water all the time. As you begin to metamorphose and move on to land, the amount of ammonia excretion is going to drop significantly, and you're going to switch into urea excretion. And also, what you should know, I said that there were only 4 nitrogenous excretory compounds: ammonia, urea, uric acid, and guanine. However, those are the most common ones. There are a whole host of others, so just be aware of there are more than just the 4 I started with. Typically in physiology, we see complexity increasing as we move up the phyla, and if an earlier phyla has a successful adaptation, we see that expressed in later phyla. However, if you look at this slide here and we look at the patterns of excretion according to phyla, we can see that there's not really any real patterns. So mammals and birds are very evolved, but mammals excrete urea and birds excrete uric acid. Reptiles are lower down, excreting uric acid. But then we've got elasmobranchs and amphibians excreting urea, but then we've got teleost fish, which are high order vertebrates, excreting ammonia. So it's quite clear when you look at this slide that the mode of nitrogenous excretion adopted by an organism is defined by its normal habitat rather than any phylogenetic evolutionary relationship. And just to really ram the message home, here we have a series of tortoises, and we can see that the tortoises who are aquatic, their major excretory product is ammonia, followed by urea. And then when we look at tortoises who live in desert conditions, their major excretory product is uric acid with very little ammonia, and then in between we have urea taking over as the main excretory product. And similarly here we have the development of a chicken and egg, so it's in an enclosed environment, and you can see this start off with this very little ammonia excreted, and that's because in the early stages of development ammonia is toxic and is going to affect its development significantly. However, as it's developed and there's more free water, we get ammonia production, but we can see that as we move through the time period, we have very little uric acid produced, and then we have significant amounts of uric acid, the amount of ammonia and urea production tailing off, and that's because the organism is trying to conserve water. Okay, and this is just a summary slide for your revision. Okay, there are 2 ways in which an organism can achieve excretion, and it should say excretion rather than exception, and these are through either active transport, so we have a waste metabolite and we actively pump it out, or through ultrafiltration, so we basically filter out the waste metabolite. Now, thinking about active transport versus ultrafiltration, I want you to think about what the advantages and disadvantages of each might be before you move on to the next slide. Now, active transport's good because it's highly selective, so pick exactly what you want to excrete out. But the problem with active transport is it costs energy, and if you've got a lot of waste metabolites, that's going to cost you a lot of energy. In contrast, ultrafiltration is good because it's non-selective. Ultrafiltration is good because it doesn't cost any energy if you're using pressure within an existing system. But the bad side is that it filters out everything that's the same size or smaller than the molecule you're trying to get rid of. So active transport's good, it's selective, but bad because it costs energy. Ultrafiltration's good because it doesn't cost energy, but bad because it filters everything. So when we look at excretory organs, we talk about nitrogenous excretory organs and excretory organs that might not will excrete another waste metabolite, but not necessarily nitrogenous. So we've got 4 types of main generalized excretory organs, and those that are mainly concerned with excreting nitrogenous waste. So we've got contractile vacuoles, which you're going to see in simple organisms, single cell organisms particularly, invertebrate nephridial organs, so we're obviously talking about lower orders, Malapigian tubules of insects, so we're getting more complicated, and then the pinnacle of excretory evolution, the vertebrate kidney. And as I said, we do have other specialized excretory organs, but they tend to excrete something other than nitrogenous waste, so they are doing waste metabolites. So we've got gills. So gills, when we look in fish, do osmotic regulation as well as nitrogenous waste excretion. Rectal glands and elasmobranchs, okay, and in the intestine in insects. So they're all will do other waste as well as nitrogenous waste. Then we look at reptiles and birds, and they've got salt glands, and that's to do with osmotic regulation. And then if you think about us with our liver, okay, our liver produces bile and gets rid of metabolic and cytotoxic waste and excretion. Now remember, porphyrin of bile is just the breakdown of red blood cells, and that's what gives our poo its nice brown colour. Alright, so just to go over the organs again, okay, so for very simple organisms which don't have a very complex metabolism, then effectively you don't need an excretory organs, and you can just rely upon diffusion. As you get a bit more complex intracellularly, you'll have contractile vacuoles. As we get further up, we've got nephridial organs, which we talk about closed and open, so protonephridia and metanephridia, and we see these in platyhelminths and annelids and mollusks. When we move on to crustacea, we see antennal glands. Insects tend to have Malapigian tubules, and then for vertebrates they'll have fully formed kidneys. Okay, so looking at contractile vacuoles, and we can think of them as basically it's really active transport / secretion. Yes, it's not individually pumping out a single ion or a single compound, it's putting them all into a vacuole and then excreting out the vacuole, but it is still a form of active transport and can work at a good scale for very simple organisms. Now, if you look at the graph on the right hand side, what you can see is that if we look at, as we decrease the salinity of water, we can see that the rate of vacuole pumping goes up quite considerably, and this indicates that for a lot of freshwater species, contractile vacuoles may be getting rid of waste metabolites, but their primary function is to get rid of excess water. So the contractile vacuoles there are working for osmoregulation, which we'll talk about in next week's lecture. Alright, we're going to jump now from contractile vacuoles. We're going to miss out nephridial organs and Malapigian tubules for a minute, and we're going to go straight to the vertebrate kidney. And the vertebrate kidney uses both ultrafiltration and active transport, so secretion and reabsorption, to get rid of nitrogenous waste products. Okay, here we have a cross section through a kidney, okay, and we can see we've got an outer condex and an inner medulla, and what we can see is we've got an outer zone of the medulla and the inner zone of the medulla, and then what we're actually interested on is the functional unit of a kidney, which is called a nephron. So what we can see is we've got an artery that comes down to glomerus, so it's basically an arteriole which divides with inside a capsule, which is known as the Bowman's capsule, and we call that glomerus and the Bowman's capsule together the Malapigian tubule. Sorry, Malapigian body. Malapigian tubules are in insects. Okay, and this is where this is where the blood's filtered. So the blood within the glomerus is filtered, and the fluid enters the Bowman's capsule. Leading away from the Bowman's capsule, we've got the proximal convoluted tube, which goes down into the descending loop of Henle. That rises up in the ascending loop of Henle. We then go into the distal convoluted tubule, and the distal convoluted tubule then enters into the collecting duct. So the whole unit, basically from the glomerus until we hit the collecting duct, is known as a nephron, and human kidneys are made up of millions of approximately about 1 million of these in each kidney. So if we were to follow the flow of material from the bloodstream down into the bladder, okay, we would have blood entering the glomerus, okay. It's then filtered. The fluid from the blood, which is of blood but not the same with the blood, goes into the Bowman's capsule, then into the proximal convoluted tubule, proximal straight tubule, the descending limb of Henle's loop, the ascending limb of the loop of Henle, okay, into the distal convoluted tube, then into the cortical collecting duct, the medullary collecting duct, the renal pelvis, which is where all the collecting duct goes into, and then we would go to the ureter. So each kidney would have one ureter, 2 ureters, down to the bladder, and then from the bladder out of the body through the urethra. Okay, so how does filtration occur? So before you move on, I want you to pause the slide and think about, okay, if we wanted to filter a solution, okay, we have a filter paper, we put it in a funnel, we pour a solution in, okay, and the clean water drips out at the bottom of our funnel. But there we're using gravity as a system to drive our filtration. So how do we drive filtration in the human body or in any organism? So how would you need a force to push to literally pull or drag the fluid through your filter. Okay, so how are we going to do that in the body? So I want you to pause the recording now and think about what force we're going to use to generate filtration in the body. So pause now. Alright, you've paused and hopefully you've thought about it, and the answer is going to be blood pressure. Alright, so the pressure of blood is going to be the force that drives filtration. So here we have our Malpighian body, which is composed of our glomerus and our Bowman's capsule. And the glomerus is, just as I've already said, arteriole, which is multiply divided up. Okay, and if we look in that, we've basically got a series of 3 different pressures going on. We've got the blood pressure in the glomerular capillary. We've got the colloid osmotic pressure, so that's because if there's an osmotic difference between the fluid that's produced in the blood's going to contain more molecules, so it's going to want to move back in. And then we've also got filtration pressure. So if you've got a hose pipe and you've squirted against your hand, yes, some drops down, but some squirts back. Right? So long as the blood pressure is greater than the osmotic pressure and the filtration pressure, we'll always produce filtrate. Remember, filtrate is of blood, but not the same with blood. And basically, what the Bowman's capsule acts, we can see here we've got those podocytes, and it's basically like a giant sieve or a colander. Okay, so everything that's less than 70,000 molecular weight or 10,000 Daltons is filtered out. So this means that glucose, salts, simple iron, stuff like that, as well as our urea, is filtered out. So this has some implications, alright? So proteins, large stuff will be left behind, but anything that's small gets filtered out and is going to enter the Bowman's capsule and then go into the proximal convoluted tubule. And basically, in adult human, we're filtering our blood, so that's about 5.5 litres every half hour. So every half hour, we're going to be producing roughly about 5.5 litres of filtrate. Okay, so if blood pressure is responsible for generating filtrate, how can we control how much filtrate we produce? Well, the simple answer to that is we can adjust blood pressure. Now, we don't want to adjust blood pressure of the whole body because that's going to have profound consequences all over, and that's a bad idea. But can we adjust blood pressure on a local scale, so particularly at the scale of the nephron? And sure enough, yes, we can. So if you look here, we've got a thing called the juxtaglomerular apparatus. And basically, it's a series of muscle cells which surround the afferent arteriole. Now, if those muscle cells contract, that means we restrict the flow into the glomerus, which means the pressure within the glomerus drops, meaning we produce less filtrate. However, if we want to produce more filtrate, we can relax those juxtaglomerular cells so that afferent arteriole opens up, there's more blood flowing into the glomerus, so the pressure rises, we produce more filtrate. So by selectively constricting or expanding is a better word, dilating the juxtaglomerular apparatus, we can control the amount of filtrates that are produced, and we can do this both using a hormonal system and by direct stimulation. Alright, so I've said that everything that's smaller than 10,000 daltons gets excreted out into the proximal convoluted tubule. So that means we've got glucose, ions, all sorts of stuff that we want back in the body because we don't want to lose, including the 5.5 litres that we're filtering every half hour. 5.5 litres of water. So, basically, the proximal convoluted tubule absorbs most of the salts, water and glucose we want. So amino acids, bicarbonate, 80% of the stuff that's in the filtrate that we want back in the body gets actively transported back into the body in the proximal convoluted tube. Then we have the loop of Henry. In the loop of Henry, filtrate enters isoosmotic to blood, so that's 300 milliosmoles, but leaves hypoosmotic blood at 100 milliosmoles. And the loop of Henry is where we get a lot of sodium chloride reabsorbed. And also water, but that'll sound really confusing for a minute because I'm saying it's going in at 300 milliosmoles but coming out 100 milliosmoles, so you'd go, well, surely you said water's gone in. But think about it, how can you reduce the osmolarity of a solution? By not adding water. Just keep that in your head. And in the distal convoluted tube we get further sodium chloride reabsorption and we get potassium ions pumped out. Okay, and the filtrate here is hypoosmotic. I'm sure that should be hyperosmotic. No, in the distal convoluted tube it's hypoosmotic. When it enters the collecting duct, we drive it so it becomes hyperosmotic. So this slide is just to show you the active transport processes working in the proximal convoluted tube to pump material back in. Okay, what I want you to pay attention to on this slide is the right hand picture where we've got the descending limb and the ascending limb of the loop of Henle and then the collecting duct. So we note that when the filtrate enters the descending limb of the loop of Henle, it's at 300 milliosmoles, and as it goes down the descending limb we can see that the osmolarity of it increases, so it's hyperosmotic with respect to blood. However, in the ascending limb, what happens is it goes from being hyperosmotic to being hypoosmotic with respect to blood. So we go 300, 600, 900, 12 and 1400, and then suddenly we start to drop, going 1700, 400 to 100 at the top of the ascending loop of Henle. And then as we go around the distal convoluted tube, we will become even more slightly hyperosmotic, and then suddenly we get back to the osmolarity of blood, and then we start becoming hyperosmotic. So if I'm going to tell you that we're not adding water into that tube, so I'm going to say that the ascending limb and the distal convoluted tube are water impermeable, so that means I can't get water in or out. How can I be making something hypoosmotic if I'm not adding water? And the answer you want to think about is simply, I'm pumping salt out. So if I pump salt out, I reduce the osmolarity of the solution, and that's exactly what's going on in the ascending limb of the loop of Henle. Now if I'm pumping salt out and I'm pumping it into the medulla, what am I going to be generating in the medulla? Well, I'm going to be generating a high concentration of ions, so the medulla itself is going to have a high osmolarity. Now if the descending limb and the limb of the collecting duct are water permeable, that means water is going to want to move out of the descending limb and the medullary collecting duct into the medulla, where it can be reabsorbed back into the blood. How do we get the salt out of the ascending limb? Okay, we use active transport. So by using active transport we can set up a concentration gradient in the medulla, causing water to move out of the descending limb and the collecting duct, generating hyperosmotic urine, but also making sure that we recapture most of the water that we've produced in the filtrate. So I want you to watch the How Do Kidneys Work flash, okay? And as you're watching it, I want you to think about, well, what's going to happen if I make the limbs of the loop of Henle much longer? What's that going to do for the amount of water I reabsorb? And I want you to answer that question in your head once you've watched the flash before you move on to the next section. Okay, so we've got a slide here which says, but what about the water and how we deal with reabsorb the water? We don't drink 14 litres of water every hour to replace the filtrate. So we've already answered this really, is that by actively transporting, it's actually chloride ions and sodium follows it passively in the ascending loop of Henle, okay? And then the distal convoluted tubule, we can generate a salt concentration in the medulla, and so that any water that's in the descending limb, okay, and the collecting duct, which is water permeable, will pass out from it into the medulla and be reabsorbed. So what we're doing is we're using a combination of osmosis or osmotic gradient, which is generated through active transport, and a counter-current flow system to ensure that the body retains water. Now this combination of active transport, okay, and a counter-current flow system is fundamentally important in physiology. And we call this combination of active transport and a counter-current flow a counter-current multiplier. And what a counter-current multiplier allows us to do in physiology is maintain a gradient and across a very high differences. So you can see at the top here we've got 300 milliosmoles, at the bottom we're at 1400 milliosmoles. So there's a big difference here. But if we look in between our descending and our ascending limb, we've always got a 200 milliosmole difference. So what this allows us to do is it allows us to generate massive gradients and maintain a fixed difference between those gradients so we can reabsorb whatever we want. And we see this in vertebrate kidneys, but we also see this in fish swim bladders, which we'll talk about in a future lecture. Alright, the trick to thinking about this and remembering this is that remember that the ascending limb is water impermeable but pumps out chloride ions which salt follows passively. The descending limb is water permeable. By pumping out the chloride ions which the sodium follows passively, we generate a gradient within the medulla. Okay, the bigger the medulla, the bigger the gradient. Okay, this gradient is going to suck water out of the descending limb and out of the collecting duct. Okay, so if we turn on chloride excretion in the ascending limb, okay, then what we're going to do is we're going to generate a bigger gradient and we're going to pour more water out. We want to stop water reabsorption. We can either make the descending limb and the collecting duct less water permeable, which is what we actually do with a antidiuretic hormone, okay, or we can switch off, we can stop pumping out so much salt. Alright, I want you to watch the flash telling you about how the countercurrent, the flash called the countercurrent multiplier, just so you understand how the countercurrent multiplier arises and how it's maintained. So what happens if we increase the length of the medulla, okay, or effectively the length of the nephron? We increased our amount of water reabsorption. So here we can see a laboratory rat, a Mongolian gerbil and a sand rat, and you can see as we move from the environment where there's plentiful of water to an environment which is very arid and there's very little water, the length of the medulla increases and therefore the length of the nephron increases as well. Okay, so we're producing, the greater the length of our nephron, the more hyperosmotic urine we can produce. Okay, and if you just watch these two animations and then just move on to the next slide, please. Okay, two things I want you to note on this plot. Okay, first of all, that according to where you live, your medullary thickness increases. So, for example, if you're a desert or an arid organism, you're going to have a very big medulla. It's going to be less so if you're mesic, so that means where water is plentiful, and if you're aquatic, it's going to be quite short. But also, there's a relationship between body weight and the length of your medulla. So, small organisms have a relatively thick medulla, large organisms have a relatively thinner medulla. And thinking about Rubner's law and thinking about the ratio of surface area to volume, why do you think smaller animals need to conserve water much more than larger animals? And you should be able to answer that, especially if you think about mammals, for example, and how they might have to deal or cope with water loss. Okay, and this is just a summary slide for you. Alright, this is just an aside and you should be able to answer this for yourself. Considering we've done the practical where we used AMREP, where we looked at filtration rates using AMREP. So I want to look at your renal clearance rate or your filtration rate in the human body to check whether or not your kidney is working properly. How am I going to do it in hospital? And what we do is we basically inject you with a known amount of inulin, and inulin is a carbohydrate which we don't absorb, so it'll just be filtered out by the kidney. So if I know how much inulin I've injected you with, okay, and I've worked out what your approximate blood volume is by looking at your size and all the rest, okay, then I can then look at how much inulin is excreted into the urine, and then I can work out, well, I know a standard filtration rate should be this, and I can determine whether your filtration rate is equivalent to that or greater than that. If it's significantly greater than that, then I'm going to know that you've got some kidney damage going on. Okay, we've talked about kidneys and we've talked about contractile vacuoles, so we're now going to move back and we're going to look at the malapighian tubules of insects. Now, these are actually really quite clever. Alright, so they're blind ending tubes, okay, they're derived from the intestine, and you saw them when we did the cricket dissection, okay, so those were the bits with the pink in. And what they do is they use active transport, okay, to pump loads of potassium ions, okay, into the tubule. So 60 to 30 times greater concentration than the hemolymph. Because there's an increased iron concentration in these Malpighian tubules, water and other waste metabolites are going to drift across. And remember, proteins aren't going to go through the plasma membrane, but anything that's small that can get across the plasma membrane is going to move into the hind gut. Alright, so this is solely like it's using active transport to set up a gradient to pull what we don't want across, but also we're pulling across stuff that we do want, particularly water and for example, say glucose or something like that might be dragged across. Okay, what then happens is that water, ions and desired molecules are then reabsorbed, okay, in the hind gut or rectum, leaving behind the waste metabolites which we don't want. And if you look at this slide here, you can see that there's that salts are being dragged out into the rectum. So we've fed locusts saline or fed them water, and if we look at the concentration or the osmotic pressure in the rectal fluid, so basically the solute concentration, we can see when we give them saline, that solute concentration goes up massively. Okay, the blood has gone up a little bit, the osmotic pressure, but not that much at all. Okay, so these are very efficient. This is a very efficient way of excreting waste metabolites and unwanted products. Okay, so you've excreted, used the Malpighian tubules to cause water and the waste metabolites to be drawn into the lumen of your intestine, but how do you get that water back into your body? So I said, well, it happens in the rectal papillary, but how? And how come you don't drag in back what you want? So what you've got is you've got rectal cuticle, okay, which is just water permeable but impermeable to everything else, and then you have this intracellular space between basically your hemolymph and the rectal cuticle. If we actively transport stuff from our hemolymph into that intracellular space, okay, we're going to generate a high concentration gradient. Because we've got a high concentration gradient in there, okay, because we've actively transported stuff in, that's going to draw water out of our intestine. So if you think about it, and because we've got our rectal cuticle as only permeable to water than nothing else, we're going to suck up all that water we've removed from our intestine, we move it back into our blood, and then actively transport our ions back into that intracellular space. It's pretty clever. Okay, this is a summary slide for you. Alright, last thing we've got to look at are invertebrate nephridial organs. Okay, and I talked about protonephridia and metanephridia. So protonephridia are the simple ones closed, okay, and metanephridia are the open ones. And if we look here, we've got a platyhelminth or a flatworm, and we've got these flame bulbs or flame cells, and we can see they're full of cilia, and these cilia are going to move, and we've got fluid moving into these cilia, and we've got these fenestrations or slits, okay, in this wall. So, okay, platyhelminths are going to be an open circulatory system, so they're not closed, so it's a low pressure system. So what can we, they're going to use filtration, but what can we use to generate a pressure to filter out what we want? And basically, they've got cilia. What can cilia do? Well, cilia can beat. If I beat, I can generate a current flow. If I'm generating a current flow, then I'm generating pressure. And this is exactly what we see with these flame bulbs. Okay, they're generating a pressure differential in them, so that fluid is pressed up against the cell wall. Stuff that's smaller than the slits or pores in the cell wall will be pushed out externally. Now here we have metanephridia. So this is where we get more complicated, and we have the system joined up, and what we actually have is we have both filtration and reabsorption occurring. So in protonephridia we would just say filtration, okay. In metanephridia we have filtration and active reabsorption going on. And if we get if we move up a bit of order as well, or upper phyla, we can even get a little more complicated, so we begin to resemble something that actually begins to look like a kidney. So you can see here we're moving towards a kidney where we have filtration or ultrafiltration and reabsorption going on, and active transport obviously. So here we have a crustacean antennule gland, and this is, we're almost actually getting towards what we can see as a normal urinary excretory system. And as we move up the phyla to mollusks, okay, and to a very high order mollusks, so we're talking about cuttlefish and octopus, we can actually see what we see is proper filtration and reabsorption, or what we call pericardial ultrafiltration. Okay, so what we've got is we've got our heart actually driving a blood pressure, causing filtration to occur, and what we would say is that this is actually a proto kidney. This is what we would consider to be a true kidney, or the derivative of the true vertebrate kidney.